Today, I will talk about our work on variant effect prediction for human proteins. And as in the introduction told, I work at Van Yüzüncü University. So let's start with a bit of motivation. As you all know, amount of sequence data is increasing, but unfortunately, amount of information that we can extract from this sequence data is not keeping up the pace. So we need low-cost, fast methods to interpret variant effects or understand variant effects. And in this sense, computational mutational effect prediction algorithms can be some good venues to explore. And in this talk, I will talk about our exploration basically. Okay, since this is a variant effect seminar, I don't think I have to introduce the problem, but I will just talk about the part that I will touch basically. In this talk, I will talk about substitutions, and I will divide them into two groups, like single point substitutions and multiple point substitutions. And then I will talk about some deletion effects, predicting deletion effects basically with our algorithm. Again, I will divide the deletion effects also into two groups, like single point deletions versus multiple point deletions. So outline of my talk will be basically similar to a paper. I will introduce the materials that we used for testing, for understanding, etc. I will introduce our algorithm PRESCOTT methods, and then I will present substitution effect predictions results, and I will present the deletion effect results. At the end of my talk, I will introduce our software and the data that we generated in this project so that everybody can access them or check them. My hope is actually, even though the focus of this talk is human variants, I may touch or might touch the effect of mutations in viruses at the end of my talk also. So I hope that it might be helpful for example, for researchers like Tim as well. The data set that we used is quite well known actually. For substitutions, we used version 0.1 of substitutions in protein gene. It had 72 proteins from various organisms, human, other eukaryotes, prokaryotes, and other viruses as well. In total, there were 87 experiments, 76 of them were single point mutations, and 11 of them were multiple point mutations. And in total, we had about 1.5 million measurements. For deletions, again, we used protein gene indels dataset, but we separated deletions from insertions. And in that data set as well, we have 60 experiments and about 5,000 deletions. The second data set comes from human proteins and ClinVar. It's about 2,000 proteins, approximately 8,000 pathogenic ClinVar labels and 9,000 benign labels. So in many variant effect research projects, there are three sources of information, and the same was true for us as well. The first source of information is sequences, and we use this information, or the source of this information is typically an MSA file in any format. The second source of information is structures from mostly PDB files or CIF files. And the third source of information comes from population. And in our case, the source of population information was NOMAD. If we use sequence and structure only, we call our method PRESCOTT. But if we incorporate population information, we call our method PRESCOTT. And now I will introduce our PRESCOTT method. Basically, in PRESCOTT method, we have two components. The first component is independent component, and the second component is epistatic component. To predict the effect from X to Y at column I, we do a linear combination of independent component plus epistatic component. We give 40% weight to independent component, and we give 60% weight to epistatic component. Okay, Now let me introduce my components. The first component, as I said, is independent component, and the basic idea is to count the frequency of an amino acid at column I. If amino acid is frequent, it means that it's less impactful. Mutation to that amino acid is less impactful, and if it is rare, it's high. It has high impact. But instead of counting 19 amino acids here, we are just grouping them into similar groups like physicochemical features. And we call each grouping an alphabet. We use a specific alphabet that you can find in our paper. This is independence component. This epistatic component. Before starting, details of epistatic component, I would like to pose a question. Okay, the question is, okay, how do we determine sensitivity of a position at, let's say, position I to mutations in a protein? The classical way, of course, is to check conservation. If it's highly conserved, it's sensitive to mutations. If not, okay, it's not. But here we are not taking the classical approach. We are using a score that we called max score. Max score, as you can also see, is a composite score. It's made up of 3 components. The first one is TJET. TJET plays the role of evolutionary conservation, but it is definitely not Shannon entropy. As I said, it's coming from joint evolutionary trees, but it plays the role of evolutionary conservation. The second component is physicochemical features of amino acids. And the third component is a structural feature called circular variance. Circular variance can be thought of as a kind of density around an atom or an amino acid, density of atoms. And it's a structural feature coming from PDB or CIF files. So we are building binary linear combinations here, TJET plus PC divided by 2, TJET plus CV divided by 2, and then we select the maximal of these. And this gives us max score for that position in general. So we define importance of a position with max score instead of going to conservation standard way. Then we have an MSA file as our input. And in our MSA file, the query sequence is the sequence that we wonder mutational effects. And we have several subject sequences. I am showing just one example. We compare our subject to query, and we find the locations that are not identical to the query, which are shown here as red. And we sum up all max scores for these positions only. And we do this for all subject sequences in our multiple sequence alignment file. At the end of the day, we have an evolutionary distance matrix, and we will use this evolutionary distance matrix to understand epistatic effect. So here comes the epistatic part. Let's assume that we are checking the mutation impact of mutation from X to Y. We select a subset of subject sequences containing Y, and then we find the sequence with minimal evolutionary distance. This constitutes our epistatic component. And again, we are going to the beginning. We have independent component and epistatic components combined in a linear fashion, and we put weights or normalize our predicted effect here with max score, which means that you give much more importance to the positions that are that have high max score. So this is our PRESCOTT score. PRESCOTT score, as I said, uses multiple sequence alignment information and structural information and physicochemical features as well. How to add population information here? As I said, our source of population information is nomad files or allele frequencies in nomad files. Basically, we rank sort our PRESCOTT scores. The purpose is just to bring them to a scale of 0 to 1 for the rank sorting, and 1 means high effect, 0 means low effect. The population part is the second term here. F is the frequency of that allele. and FC is a cutoff frequency. Cutoff frequency here, we use log 10 values of them, but cutoff frequency is 1 in 10,000. If it is more frequent than that, Heaviside step functions, the function tells us, okay, this term should be active, which means it becomes 1 if F is greater than FC, otherwise 0. C was a scaling coefficient that we tested, to understand it, but we decided to keep it 1 at the end of the day. And then FC minus F divided by FC gives a score between 0 and 1. Basic idea here in the population information is really to modulate PRESCOTT score based on its frequency. If it is too frequent, it will reduce it more. So, okay, now let's start the results. Here in the results, at least in this slide, on y-axis, we have 32 experiments from human proteins. It contains single point mutations and multiple point mutations. And on x-axis, we have Spearman correlation. So, and we have 4 methods. PRESCOTT is published in Genome Biology, recently published in Genome Biology, and I am sure almost everybody here is aware of the other 3 methods as well, alpha missense, ESM1B, and EVE. In general, we can see that, okay, on the top right part, there are some proteins that almost all methods predict quite well. On the other hand, on the bottom left-hand side, we can see that almost all methods fail for some proteins to predict them well. But when we check the averages over these 32 proteins, we can see that PRESCOTT and alpha missense have the same average Spearman correlation, 0.460. And then E follows them, and ESM1B is the 4th one. Here I am presenting only the 4 of them, but we wanted to see our results in a bigger context, I would say, and we compared it with about other 30 methods. Again, alpha missense and ESM1B have the same score, but the third method is iGEM. iGEM is basically PRESCOTT without physical, chemical, and structural information, and then transceptive family and POET follows these methods, and it goes on like that. As I said, okay, my data set that I have just presented in 2 slides before contains both single and multiple point mutations, and I wanted to show you its performance for only single point mutations and for only multiple point mutations as well. When we check for only single point mutations, we can see that alpha missense is slightly better than PRESCOTT. But when we do multiple point mutations, PRESCOTT is better than alpha missense as well, and then EVE follows them. The second data set that we use to understand, or let's say check our program basically, was human proteins and ClinVar labels. As I said, we have about 2,000 proteins and 17,000 variants, approximately divided into equal 2 groups, pathogenic and variant. On left-hand side, you can see their distributions, score distributions for pathogenic and for benign mutations. On the right-hand side, you are seeing AUC curves and AUC scores. As you see, PRESCOTT score, AUC for PRESCOTT score is 0.88. But when we include population information here, we can see that PRESCOTT score improves and becomes 0.95. It becomes PRESCOTT score basically. And alpha missense score is 0.91. And then even ESM1B, at least for this data set, were generating quite similar results, 0.87. Okay, up to this point, I talked about only substitutions, multiple and single point substitutions, but PRESCOTT algorithm can be used for predicting deletion effects as well. On x-axis, you are seeing about 60 experiments from different species. It's not just human here. And on y-axis, you are seeing about 20 methods predicting deletion effects. The top row is PRESCOTT results. And my colors are Spearman correlation, but pay attention that it is between 1 and -1. So if it is dark red, it's good. If it is dark blue as well, it is good. But just the scale is reverted, I would say. So when we check it, PRESCOTT results are mostly red, but for some proteins here, for example, if you can see my cursor, TNKS2, the predictions are not good. But the important point here, similar to human mutations or experiments, for TNKS, the predictions are not good by any method. We don't know if it is specific to anything specific to oligomerization state or any other problem, but it's not good in almost all methods. When we average this whole row to obtain an average Spearman correlation per method, we will obtain something like this. And in terms of average, our PRESCOTT results are the best, 0.606. And then program family results comes after them. Program 2B and I think program 2M follows it for single point deletion effect predictions. Unfortunately, for multiple point deletion effect prediction, we do not have too many experiments, but still I wanted to present my results for this part as well. Okay, we have 3 experiments, and again, the first row is PRESCOTT results. And I do follow the same protocol, basically get the average of the 3 experiments to obtain a kind of per method score. So when we do per method score, 0.476 is our average Spearman correlation over these 3 experiments. And then I think it was transception non-retrieval methods were the second best for multiple point deletion effect predictions. So I want to summarize our findings, and as I've just shown, PRESCOTT is quite good for predicting variant effects in human proteins, but I should really emphasize that we did not develop PRESCOTT to predict human proteins well. We developed the method as a general method to predict mutational effects for all species, I would say. We tested it, for example, for some viruses, and we have a preprint here. And the results for viruses are also quite good. Here we see also introducing physicochemical features, structural features, and population information can improve variant effect predictions. And the same algorithm can be used for deletion effects as well. But sometimes you may have to tune or change important schemas to predict deletion effects. Before finishing, I would like to introduce our webpage. Our webpage, you can access the webpage basically with the QR code here. It has 3 parts. First is on the left-hand side, PRESCOTT web server. In the middle, you can see PRESCOTT web server, and on the right-hand side, you can see PRESCOTT database. PRESCOTT database of, let's say, 19,000 human proteins. When you go to PRESCOTT web server, you have to put an MSA file in FASTA format as input, and you have to input. You have to use an input of PDB structure of your proteins of protein of interest. A good way to obtain both of them in one shot would be to use collabfold, but one has to be careful because in collabfold, the multiple sequence alignment files are given in A3M format. You have to just convert them to FASTA format and then remove the gap columns. And in this way, you can obtain, you can calculate variant effects for your own protein. And here I want to show our PRESCOTT database as well. As I said, it contains about 19,000 human proteins, predictions for all of them pre-calculated. If you go to the PRESCOTT database, you will encounter this screen. In our PRESCOTT database, we use 2 values. To search for the proteins of interest. It can be your UniProt ID of your protein, or it can be name. But as you all know, names may not be unique, or there may be some, multiple names for a protein. So I would suggest you to use UniProt ID if you have your own protein. And when you do this, you will encounter an interactive map like this. You can slide it to left and right to focus on it, and you can get screenshots, or you can download all the data as a zip file for your protein. Our source code is also publicly accessible with GPL version 3 license. Sometimes we may need to calculate variant effects for, I would say, many proteins. And so we prepared the Docker image, and you can use the Docker image to calculate in batch mode, I would say. And I would like to finish my talk by thanking to Alessandra Carbone. She was my mentor at Sorbonne University. I would like to thank Thomas Henry. He was like my commenter, and he really helped us in this project with his experimental insights. It really shaped our project, I would say, significantly. I would like to thank Laurent David. He designed the web page and the database. And our sponsor, of course, ANR. ANR is equivalent of NSF in the US, National Research Agency of France. And I would like to thank French supercomputing infrastructure to make this research for giving us the computational resources. And in particular, Janzai Supercomputing Center helped us a lot. And I would like to thank you guys for your attention and for your invitation as well. If you have some questions, I can happily answer.
Thank you for a great talk. Let's see if we have any questions in the chat box. Okay, we have one. Okay.
Sure.
So did you see declining performance of PRESCOTT or PRESCOTT with shallow MSA input? And at what depths does performance drop off?
Okay, decline from PRESCOTT for shallow. No, I mean, like, that's an interesting point. I mean, it comes up in almost every research related to this kind of, I mean, like using MSAs, but somehow we could not observe any, I mean, like significant correlation between MSA depth and our predictions. I don't know, maybe it may be important for, I mean, for different methods, but in our case, no.
Okay.
I mean, like I can give you some particular examples, like in, especially for viruses, in one MSA, we had about 200 sequences. It was giving 0.6 Spearman correlation, and we had about 10,000, and the Spearman correlation was 0.2.
Right, that's actually quite surprising. Yeah.
Yes, yes, it is interesting. I mean, we also checked it a lot, but yeah.
I think I'll do the second question. So in your model, when you present the loss function, you use a linear combination of the independent and the epistasis component. And you set the number to be 40%, like 60%. Is there any like clear reason that you choose the number? Or like you do like some cross validation or like these type, like try and error?
We did a kind of like separated data sets like into two groups, into independent groups, and then check the I mean, like if giving equal weights or just reversing the weights. But somehow, I mean, this one actually we tested, I think, in 3 independent data sets. We separated our data set into 4 groups, and in each group we checked it. Somehow it was consistent, really. I mean, I do not claim that in one case it is 61 point blah blah or 40 or 39 point, but this was approximately giving us the best scheme. I mean, giving more weight to the epistatic component and then less weight to the independent component was working us for the best.